Good morning, dear students. Today we are going to start a new chapter, that is DNA repair. That means DNA are having some damage and that need to be repaired. Okay. So first we need to think about the complexity of our genome. Human genome. I think the genome, that term is clear to you. Anyway, I'm not going to means explain that term now. Just understand that our human genome contains how many base pairs? 6.4 into 10 raised to 9 base pairs are present. Look to that figure. 10 raised to 9 means after 1 there will be 9 zeros. That much huge figure. So that much base pairs are there. Its correct incorporation is required here. So during the incorporation of these bases to a daughter DNA during replication, there is chance to have some errors even if proofreading mechanisms are there. So because of that, our cell possesses a DNA repair system. And this DNA repair system is highly efficient. Efficient to reduce the chances of errors incorporated to DNA. And what is the rate in the presence of a DNA repair system? That DNA repair system can reduce the rate of error to 1 per 10 raised to 8 nucleotides. That means if we consider 10 raised to 8 nucleotides, out of that only 1 will get mistakenly or wrongly incorporated to the DNA. So, to that much level, it is reduced because of the DNA repair mechanism. And we need to understand that how these errors are happening to the DNA. A lot of reasons are there, and main reason is if you are exposed to ionizing radiations or because of the presence of certain reactive chemicals. And from where these reactive chemicals are forming in our body, that may be produced because of our cells own metabolism. Okay, so among different reason, these two are important reason that cause DNA damage. And if we take a warm-blooded animal, because of erroneous incorporation during DNA replication, how many bases are losing per day? 10,000 bases are losing. We know that from our DNA during replication and forming the daughter strand, that time so many nucleotides are incorporating to daughter DNA, and that time if any misincorporation happened, that misincorporation need to be removed. And by that way, if we take the case of a warm-blooded animal, in one day it lost 10,000 bases. Okay, then the situation become more serious when if that errors happen to germ cells. If it happen to germ cell, then that error may transfer to the next generation. It's fatal. So, it's very important to understand about the DNA repair, and it is very important to have a proper DNA repair system inside each and every cell. So, different methods also there for repairing the DNA. Let's see one by one. Direct DNA repair is there, and the direct DNA repair itself are of two types. They are photoreactivation and methylation repair of DNA. And that methylation is caused by some alkylating agents. Okay. Then second type of repair is methyl mismatch repair. It is otherwise called MMR. Then base excision repair, it is otherwise called uh, BER. Then next one, nucleotide excision repair. It is otherwise called NER. And the next one is double strand break repair. Double strand break repair, that also is of two types: homologous recombination repair and non-homologous recombination repair. And look the difference. Here it is mentioned that a double strand break. That means all the other methods are handling single strand break. This method of repair is handling double strand break. Okay, in today's video, we will learn about only one method, that is direct DNA repair, that itself are of two types: photoreactivation and methylation repair of DNA. Okay, let's see the direct DNA repair methods. And in that, the first one is photoreactivation. Photo reactivation. Photo means something relating with light. That means light is helping to correct that DNA damage. And this method of DNA repair is present in the case of both the prokaryotes and eukaryotes. Okay, and so we can understand what is the process. And before that, I'm going to show you an intact DNA. Here we can see it's very correct, a perfect DNA. Then how that perfect DNA changed to a damaged one as it got hit by UV photons. So UV photon is this one. It came and it hit here. It usually affects pyrimidines. It usually affects pyrimidines. We know thymine is a pyrimidine. Most commonly, this damage is affecting pyrimidines. Here it affects the thymine. After it hit there, instead of pairing with the adenine in the opposite strand, these two thymine, they pair each other and form a dimer. So that is the problem. As the dimer is formed here, it causes a bulging of this portion of the DNA, and also it will hinder or halt all the transcription and the replication process. Then we should know that how this dimerization happened here. Dimerization between this thymine base. So that's what I'm going to show you. Uh, this is one timer and this is another thymine. And in this structure, we can see double bond. One double bond here for this thymine, and another also we can see. So as the photon hit the thymine, this unsaturation got changed to saturation as it got energy. So by that way, that double bond can get saturated to form a cyclobutane ring. So that's it. Form a cyclobutane ring. That ring we can see. So that's the problem. We need to correct that problem. Only then it is possible to bring about transcription or replication for this DNA. Okay. So let's see that. Okay, these dimers will block DNA replication and transcription that I already told you. Then which enzyme is helping to remove or correct that damage? That enzyme is DNA photolase. Okay, and we need to understand some points about this enzyme. This DNA photolase is a flavor protein, and also it has two light harvesting cofactors in it. Okay, so which are those cofactors which can harvest light? from the name of that enzyme itself, it is clear that it needs the presence of light for its action. So, light is harvested with the help of two cofactors that present inside this enzyme. They are FAD and MTHF. FAD means flavin adenine dinucleotide and MTHF is methynyl tetrahydrofolate. So if you feel that it's difficult to memorize this much expansion of these cofactors, let's keep in your mind only this short form FAD and MTHF. Okay. Okay. So next we are going to see the process. This enzyme has a peculiar feature. Peculiar feature is that even if it needs light for its action, it can bind to the damaged DNA only in the dark. It can bind only in the dark. Okay, so now it is dark and that enzyme come and bind that damaged site. And we can see the two cofactors here, MTHF and FAD. And in the presence of light, this enzyme got activated. As it activated, the one light harvesting cofactor, that is MTHF, present in it absorbs, it takes high energy electron from the visible spectrum. It takes electrons or energy from the visible spectrum. Visible spectrum, and then it transduces that energy and electrons to the other cofactor, that is FAD. And as FAD receives that electron, if it is reduced to FADH2. And from this FADH2, this high energy electrons go and hit this thymine dimers and remove that cyclobutane ring, that dimer, and freed that thymine. By that way, again, it will base pair with the bases that present in the opposite strand. In that way, that repair is correct. Sorry, that damage is corrected. So now, we can see here that damage is correct with the help of DNA photolysis. Next, we are going to see the second direct DNA repair method. That is there, the damages happen because of some unwanted methylation in some regions of the DNA. Unwanted methylation. Who or what make that unwanted methylation? Some alkylating agents, they give or they produce that unwanted methylation in the DNA. So that also is a damage because that methylation happens in some unwanted sites. Let's take an example for such type of unwanted methylation. Look, in this picture, we took guanine as example. This is the picture of normal guanine. And to this guanine, because of the action of some alkylating agent, one methyl group got added to the oxygen in its sixth position. Where? To the sixth position. Oxygen in the sixth position got an addition of methyl group, and by that way that guanine get converted to O6-methyl guanine. That is a modified form, methylated, unwantedly methylated guanine. It is not desirable for a normal DNA. Okay, need to correct it. So there come the role of an enzyme. That enzyme, what it need to do? It need to remove this methyl group and need to convert back that O6 methyl guanine to the normal guanine. So, which is the enzyme? And before that, we need to understand that what's the problem? What's the problem that cell faces if such methylation happen? Or some altered that this type of alteration happen to some base. As we took guanine as example here, I will show you what will happen here. As normally in DNA, guanine pairs with cytosine. Okay. So as this guanine get modified through got added a methyl group, it become O6 methyl guanine. In this form, it has affinity to pair with thymine instead of this cytosine. So normally guanine need to pair with the cytosine, but as it got modified because of the addition of one methyl group or at the oxygen at the sixth position, it prefers thymine to form base pair. So, in the second round of DNA replication, if it happen, then what will get incorporated to that DNA strand to against to that thymine again, and adenine will get incorporated. So in that way, that mistake will continue after one round, after several rounds of replication. That mistake will sustain there and it will lead to serious problems. So need to avoid that. So that is a complication. So let's see which is the enzyme that is removing this methyl group. Which is the enzyme? Two enzymes are here. First one, methyl guanine methyl transferase. It is otherwise called MGMT. Okay, MGMT. And which gene is encoding this enzyme? That is ADA gene. This is the gene, ADA gene. It synthesizes this enzyme to correct or to change or to remove the methyl group from the bases. Mistakenly incorporated methyl groups are removed by the help of this enzyme. One more enzyme is there. It is ALKBH. And this ALKBH is an alpha Kg deoxygenase type enzyme. And how these enzymes correct this methylation? Through a process called oxidative demethylation. Oxidative demethylation means what? By oxidizing certain atoms in this structure, it removes the methyl group added or attached with that molecule. By oxidizing it, it removes the methyl group attached to that molecule. So that process is oxidative demethylation, removing the methyl group. So these are the enzymes. So in today's section, in this video, we have seen a general first overview about which are the different types of DNA repair methods. Then we learned about the direct repair methods like photo reactivation and methylation of DNA by alkylating agents, how it is corrected. So today we can wind up this section. Thank you.
I'm an icon, man. I'm a girl about a 10. I should fuck her in the Benz.